Okay, Bismillahirrahmanirrahim. Assalamualaikum and good morning. So, I thought for today I will explain few things about there's a few important parts and components in this paper. So, dalam author tu saya dah upload. Saya student dah dapat access kan. So if you can access, so you will have a copy of this paper. Otherwise, if you don't, you're not able to access, you always can search this paper. Alright. There are two things that I would like to cover in this lecture. One is on the high resolution imaging of biomolecular binding studies. And then the second part is actually how we prepare the sample. The biological sample has been prepared so that we can conduct the imaging. So there is two parts in this one lecture. So I strongly suggest that the student should take notes. Okay, you dengar my lecture online ni. So at the same time, you grab a pen and paper, so write down what I said. So because I will highlight the important points, so that when come to the revision, it will be easier for you to pick up which point I have highlighted. Because the one that I highlight for you is really important for you to know. Okay. So high resolution imaging is called, but the system name is actually known as surface plasmon microscope. Alright. In this paper, we will look at two types. One is known as wide field surface plasmon microscope, and the other one is the standard one. Alright. The standard one is the normal basic surface plasmon, what we call. So we call it SPR. Only abbreviation is SPR. So if it's wide field, then we call it wide field surface plasmon microscope. Why? Because wide field enables you a greater or larger, bigger area for you to examine on the surface. Later we will look at the process of imaging. Right. Surface plasmon microscope is mainly built around the Crashman configuration. So I will show you what is the Crashman configuration later. So in this system, what we do is we obtain the image by looking at the intensity map of the laser beam which is reflected from the prism. Okay, this is dependent on the light source that we use. In surface plasma, we use P polarized light. Okay, P polarized light, and the resolution is limited to few microns only when you use the standard one. But after certain research and certain improvement, when we come up with the wide field surface plasmon microscope, so here it enables you imaging at higher lateral resolution. That means better lateral. So lateral means in this direction, right? Lateral resolution. So if not, you are looking at vertical. So we are not concentrating on vertical, it's on the lateral resolution. Then compared with the prism base, because the prism base is the standard one. The wide field one, we use objective lens like in the microscope. So in this study particularly, we will look at the patterning of extracellular matrix protein and the specific antibodies. This is one type of application which is quite good for you to understand. So at least you know one particular biological studies, as in this subject biomedical engineering, you are able to understand and explain about at least one application. So this is called molecular binding and extracellular matrix proteins antibody based studies. Right. So moving on, what is surface plasmon? So surface plasmon is the oscillation of free electrons. Okay, oscillation of free electrons. So how these free electrons, because we know there is a charging of positive to positive, negative, the charging of electrons on the free area. So how do you find out electrons on the prism? Because the prism is being coated with metallic conductors layers such as gold or silver. In this study, we actually use gold, right? So for the white field, we use the gold one. So and then we use P polarized light, striking the metallic coated area from a region with the specific angle theta p. So with this angle, you will be able to generate the surface plasmon resonance. That's what we call the simplified name for the collective oscillation of electromagnetic wave which propagate on the surface, right? But the problem is standard Crashman configuration surface plasmon. What happens is the energy decays on the surface because of the charging of electron and electrons. After a while, you will find out that there is a limitation on the prism based system, because the what we call the surface plasmon oscillation, they spread along the conductive layer. But in the wide field surface plasmon, we will have a different setting. So this one is the Crashman configuration, the prism base. Alright, so you have the metallic layer, alright, and below the metallic layer, on top of the metallic layer, we deposit the biological compound. And then you have the incident beam and then the reflected beam. This is the angle theta for excitation of surface plasmon. So the thickness of the material is around 50 nanometers. Alright, so we use this one is prism based. This is what we call Crashman configuration. Right, this is the same thing. That means there is a incident beam, reflected beam. So to show this, the surface plasmon propagate between the layers, the metallic layers. Alright, so below is the comparison of the system, that we have the objective lens and we have the cover slips, glass cover slip. So what is the difference in the Crashman configuration? The standard one is this prism, right? Here we replace the prism surface with the glass cover slip, which has the coating of the gold, 50 nanometer golds at the interface of 1 nanometers chromium on the glass. So why we use the chromium? Because when there is the presence of chromium, will make the adhesiveness of gold layer much better on the glass surface. So the thickness is 0.18 millimeters. So this is the objective lens. Between the glass and the objective lens, we have a drop of oil, which used to reduce the interference. At the same time, you have a focus beam on the glass light on the sample. So this time the surface plasmon is still the same, occurs at the metallic layers, but the setting is different because the beam direction is from the below of the sample, the molecular is on top. Alright, this area. And this is the just basically the cover slips, the settings, so you can look at the details later on. So having explained about the wide field and Crashman configuration base, there are few more details on the wide field surface plasmon system, right? So let me explain few if possible. So the difference, again I said, we use the oil immersed objective lens. So now the angle of the aperture of this objective lens is 1.45, and then it doesn't actually has the same incident beam, reflected beam, because it is now has a different setting of beam direction, but the angle theta is still the same, right? Angle theta is still the same. Now the lights the beam is actually been diffused using a diffuser, so that the multiple point of lasers focusing on the sample is achieved in the wide field surface plasmon system, right? And now you are actually imaging from the from the bottom of the sample, not same like this. So you are actually striking from top and then reflected beam. So this time you are actually imaging from the below, right? These are the few comparison part. I mean, if you read some more on the surface plasmon system, you will find out few more different what we call settings, so that you can compare the standard system with the wide field surface plasmon system, right? When I mentioned about the biological sample, so this is how you actually will create. Basically, let's say this is the glass substrate. If this is the glass substrate, how we actually can deposit materials on this glass surface. So a technique is called microcontact printing, right? This is what they call fabrication of the stem. So in this study, we used 1.8 and 3.8 micrometer wide to micrometer deep repeat gratings. So we actually created the stem itself, right? So the process is mentioned here. You can read, but I will show you with the diagram, which will be much better. Okay, firstly you have the clean template, glass template. Okay, by using this glass template, so you use silicon elastomer. That means you prepare the silicon elastomer with the ratio of 2 to 98% casting the stem. So when you pour on top of the clean template, so you let the polydimethyl silane PDMS stem setting on the template. So after a day, like an overnight, so you can peel off the PDMS stem, right? Now you have the stem, so you can actually produce the stem according to your requirement for studies. For example, here we use 1.8 and 3.8 micrometer. That means 3.8 in this direction, a bit thicker prints, and 1.8 in another direction, right? So basically what you do is you use the PDMS stem and you ink in the protein. Fibronectin is a type of protein, so you ink in the protein, extracellular matrix protein, what we call fibronectin, and then you stamp on the surface. So first print is 3.8, second print is 1.8 micrometer. That's why when you ink the stamp, so you leave it like, for example, 1 minute. So you let the protein attach on the stamp, basically, and then you stamp on the glass. So when you image on the glass, you're going to see there is protein deposition, evenly distributed monolayers of protein. So sama je macam when you use stamp kan, stamp chop nama kan, is the process is quite same. So you just take a rubber stamp and you put a stamp. Whatever you stamp, that's going to be on the paper. So here a bit different, that the presence is actually the protein molecular layer on the surface. So this one is the image produced with the normal microscope, what they call DIC, differential interference contrast microscope, with the scale bar of 20 micrometer, right? After that, we actually study the purpose of the stamping is actually we want to look at how sensitive is the surface plasmon system, the wide field surface plasmon system, right? So I'll explain a little bit on the results. So the figures in the left column, A, B, C, D, this one is on the wide field surface plasmon, and A, B, C, D on this one. The right side is the is the AFM images, atomic force microscope, right? And then this diagram is to help you understand uh, how the attachment process occurs, right? So basically what we did is we used wide field surface plasmon to image the surface that is deposited with the fibronectin. Earlier when I explained to you there is 3.8 and 1.8 micrometer. This is exactly the structure. So we have fibronectin and then the substrate. Substrate means the glass free area, right? So you have 3.8 and 1.8. That means when you stamp overlay between each other, and then you will have a center like a square. So the square is actually the free areas. So in the biology, if you look at the proteins behaviors on the surface, a protein will not attach to another protein. So the protein will find the free areas. Therefore, we use in this study fibronectin and laminin, two types of protein on the surface. So fibronectin deposited first through microcontact printing, but laminin is just poured on the surface, right? So initially we look at the contrast changes. What happened is, this is the initial image, fibronectin only. B is second, that's we have the presence of laminin. What happened is the laminin structure fill in between the fibronectin, so you are able to see there is the images, the Greek's pattern is become much more clear, because the idea of wide field surface plasmon system is to sense the changes on the molecular attachment on the surface. So the next followed by laminin, and then amplified with anti-laminin. Anti-laminin means rabbit antibodies, which is conjugated with rabbit. So anti-laminin, antibody to the laminin. So the antibody will definitely find the laminin, specifically will bind to the laminin. It will not go to the fibronectin. Therefore, you can see the patterns become much more visible. And then we also at the same time try to test whether the system are actually picking up the correct signal. So we use a mouse conjugated antibody for the fibronectin. So what happened is, this time the antibody will bind to the fibronectin protein, and then therefore you can see there is some changes on the grid patterns which is produced here. So if it is not clear on the surface plasmon, you can see there is big changes on the AFM images C and D here. So normally with the bare eyes, sometimes the what we call qualitative images, alright, qualitative measurement can be challenging. Therefore you have to extract the information from the surface, and then you plot them as a graph to show. Later I'll show you the graph. Okay, but that's the idea. We use another system like AFM to actually do a comparative analysis, same like when you do your PSM. Macam kamu buat PSM, you will compare your result with another system. If you design a system, so to show how the system is reliable, so we need another system. Therefore in this study we use AFM system, right? So when you use mouse anti-fibronectin, you will see the layers become more or less same at the level of thickness. Therefore we further highlighted the what we call anti-rabbit antibody, the laminin area, with the final secondary antibody. Alright, so when we amplify the antibody, the secondary antibody, so it shows that the contrast again increase. Right, this it shows in agreement with the results that we obtain through graph measurement. Okay, this one is just the same surface plasmon images. So if you can see, what we did was we selected the protein grid areas, the 3.1, 3.8 and 1.8 micrometer area, and then we minus it with the fibronectin free area. So there is a squares, so we minus the data of measurement. So this is how it looks. Sample looks when you look under the AFM, atomic force microscope, because atomic force microscope also has similar measuring technique with the wide field surface plasma system. However, the optical arrangement and the laser beam uh, uh, pointing on the sample is quite different. But it is good to measure the molecular thickness on surface. However, there are a few modes and setting on AFM which make it quite complicated compared to surface plasma system. But the closest system that is useful that you can use is the AFM, right? This is the graph. Okay, so you have fibronectin only, and then you have fibronectin plus laminin. When you put the antibody, the idea is to amplify the patterns. So antibody, primary antibody to the laminin, it does increase the contrast, and then it further reduce because we highlighted the fibronectin protein this time, and then again secondary anti-laminin. So this does match the findings that we measured in the images, and also the diagram representation that I showed this one. Okay, so when you try to understand that graph according to this diagram that I've shown in figure 3, you will be able to understand much better. So basically that is the idea of in this paper, to show the resolution, what we call resolution measurement, that the system are able to pick up fine structures, molecular bindings, right? And this does actually shows how sensitive is the surface plasma system to measure molecules deposited on the surface. This one, towards the end, we did the immunostaining. So immunostaining is actually to what we call to verify, to confirm that the antibody is actually function as we mentioned in the studies, so that you don't just simply test, because sometimes the antibodies that we buy from the company, they do expire. So because of they will expire, we can actually be working with the biological samples or biological material which is not working properly. So for that, you have to do the immunostaining to verify that the antibody is actually binding to the specific proteins. So I think that's all. Is there any question from the class?